people might want to use a soil health assessment. And there are those who want to use a soil health assessment as an indicator of progress. They want to know if their management is making their soil healthier. And in order to do that, we are working on a program for soil health targets, which will be described in more detail in a talk by Dr. Christine Morgan at the upcoming Tri-Societies meeting. But we're also going to talk about today a little bit more. There are others who want to use soil health assessments to quantify functional outcomes. These questions are about whether the soil is performing each or all of its jobs to the best of its ability. So we're working on quantifying specific outcomes like the ability to store or cycle carbon, cycle nutrients, store or deliver water, filter water, suppress disease and pests, act as a reservoir for biodiversity, and likely more along the way. That's kind of where we're going and what we want to be able to do with that. And so now I'm going to get into just a little bit of the data that is specific to what we did here in our project. I would say that one of the most universally accepted points from the soil health movement is that soil organic carbon is important. Increasing soil carbon is not just for the sake of storing it in the soil, but also because that carbon is useful. It is used in the soil. The soil's specific capacity to function, to do that work, tends to increase when soil organic carbon increases, which makes some sense. Most soil organisms are heterotrophic. They need carbon for food and energy. If they have food, they can do work. And so we are looking here at a list of different indicators that we measured, just a subset of them, and we see that they all have this relationship to organic carbon. First one that pops out is this really strong relationship between total nitrogen and total carbon. It's no surprise, we know that these are coupled. But then I thought it was kind of interesting that soil organic carbon and total nitrogen have the exact same degree of correlation to potassium permanganate oxidizable carbon, which is often called active carbon. And then, as has been seen in other studies, potassium permanganate oxidizable carbon has the strongest relationship to the overall soil health score as calculated by Cornell in their system. So all of the indicators have a relationship to soil organic carbon, and it's actually relatively similar to that relationship to potassium permanganate oxidizable carbon. One of the ways that we have always analyzed whether or not a measurement should be used as a soil health indicator is whether or not they are sensitive to management. And so to do that part of this assessment, we used a meta-analysis approach called log response ratios. So within each site, we paired treatments that only had one difference. For this presentation, everything else was kept the same except they used organic matter in their nutrient management strategy, either as compost, manure, or biosolids. So using this method allowed us to measure the relative difference between nutrient treatments while we controlled for inherent features, allowing us to compare the reaction among multiple sites. So when you're looking at these figures, you want to note a mark that crosses the middle line means there's no significant difference when that management has been implemented. So for all of the carbon pool or carbon flux measurements that we analyzed, they all increased with the addition of organic matter. No surprise, right? You're adding a lot of organic matter, you're going to get an increase in carbon and carbon-related pools. Similarly with nitrogen, all of the nitrogen measurements except inorganic nitrogen measured with the H3A extract were significantly increased with the addition of organic amendments. So in the story of soil health, I told earlier, soil health, soil carbon, soil functions, it seems maybe a little surprising that the overall microbial biomass using PLFA biomarkers did not show a significant difference, but we'll get back to that. A couple more of these response ratios. We tested just about every different indicator we had, and we tested every different kind of management practice using those response ratios. So here we're showing that measurements related to the physical structure and related to water did not have as consistent of a response to manure. So the methods matter. Here we see that the wet sieve aggregate stability procedure called AgStab ARS on this did show a change in stability, while the aggregate stability method in the cache test and the phone app SLAKES did not show a significant difference. Bulk density did decrease with additions of organic matter. And then using available water holding capacity using intact cores did capture a difference, while the more standard repacked method did not. And so I think that, that's something, again, we're going to come back to, this difference between the ability to see differences using the available water holding capacity using intact cores versus the repacked cores. KFS, in case you're not sure what that is, that's saturated hydraulic conductivity. There was really wide variation when looking at all of these sites together. We did not see a difference in saturated hydraulic conductivity, but often we did see differences between treatments at individual experiment levels. So in addition to responsiveness to management, we're also working on understanding inherent features, particularly related to soil forming factors and how those set a limit on the capacity of the soil to function. And so that's part of the reason that the geographic breadth of this project is so important. And so here, this map of North America is using the Hargreaves climate moisture deficit to color the map. And then you see those black dots represent our sample sites. The figure on the right is a detrended correspondence analysis of the 16S bacterial and archaeal genomic data from this project. So this is it's a multidimensional analysis and shows the two axes that explain most of the variation in this multidimensional space. The main thing to key in here is that the closer 2 points are to each other, the more similar they are. Each dot in that detrended correspondence analysis represents the entire community from one soil sample. So we see these samples from the most extreme dry climate in red, grouped over here, are very tightly bunched together. And most of the blue dots are bunched and with a gradient here in the color. And you may notice that there's a little bunch of these yellow and orange colored dots over here on the left. It may seem out of place. We checked it out. We thought that was interesting as well. Turns out those are in a dry climate, but in an irrigated system. I've seen a lot of these microbial community ordinations that demonstrate that pH is a driver of the microbial community composition. And in this assessment of our long-term agricultural research sites where pH is often managed somehow, it's no different. The color gradient changes along that x-axis, and that x-axis is what's explaining 45% of the variation. So we see that pH is is likely having a strong relationship here. And so this together, we're seeing that at the continental scale, we see that moisture and pH play a significant role in bacterial and archaeal community structure. Zooming in here a little bit, we have another detrended correspondence analysis, but now we zoomed into just one experiment in Kimberly, Idaho. It's on a silt loam with conventional tillage, diverse rotation, and no cover crops. We sampled 4 of their treatments. One that got 10 tons of dairy manure annually, one that got 30 tons of dairy manure annually, one with synthetic fertilizer, and one with no fertilizer. And while we didn't see any difference in the total biomass of the microbial community, this analysis clearly shows that there is a difference in the community structure. And so once we get to a small enough scale, we can see that the community structure changes by treatment. The axis, that x-axis down there explains 54.5% of the variation. And we see, those colors change from no fertilizer to synthetic to 10 tons to 30 tons. One more of these specific site-specific detrended correspondence analysis. Here we have an organically managed site from Michigan with sandy soil that used a diverse rotation and cover crops. And they had a factorial design using just organic fertilizer or organic fertilizer plus dairy manure compost and conventional tillage or strip tillage. And you can see by the color of the dots that adding dairy manure compost clearly caused a separation in the microbial community, but that the shapes, which represent the tillage types, are interspersed within those colors. And so they don't show that clear trend between tillage. There are a lot of other sites in the experiment where we do see a difference in the microbial community based on tillage. But here, where we have the addition of manure and we're in a sandy soil, we see that the manure is driving the community change and not the tillage. So as we're working to evaluate the measurements, we continue to think about what we need to describe the soil's capacity to function. The capacity of the soil to make nitrogen plant available is probably one of the most obvious and influential functions for many producers. And so in this trial, we don't have a absolute direct measurement of how much nitrogen the soil makes available. But there have been a lot of studies that have related potentially mineralizable nitrogen to aerobic incubations of nitrogen mineralization, as well as those that relate potentially mineralizable nitrogen to field nitrogen mineralization trials. And they found that although it's imperfect, it's a good proxy. But still, there's no commercial labs that really want to run a 7-day anaerobic incubation. And so what we did is we used forward selection regression analysis, and we tried just about every different kind of sets of indicators to see how well we could predict PMN. The most predictive set of indicators was total nitrogen, carbon dioxide respiration measured with a 24-hour burst, clay, sand, and temperature. And so using those, that multiple regression for the row crop systems, we were able to explain 72% of the variation. And in the perennial systems, we're able to explain 85%. Using just the sites that had the manure added, we could explain 86% of the PMN response with these other simple indicators. We also have a full suite of functional metagenomic data for all of our sites, which is amazing. And I can't wait to relate what we found about these indicators to the specific genes that will really help us understand more about the microbially mediated nitrogen cycle. In the arid West, the ability to store and deliver water might be even more important to producers than nitrogen. And so our team has been working on new pedotransfer functions to calculate available water holding capacity using simple measurements. The current model used by most labs, shown by the red dotted line, is insensitive to changes in carbon. But our data showed a difference in available water holding capacity using intact methods for most management changes, and that was concomitant with a change in soil organic carbon. So the new model that we've developed shows a significant difference in the capacity to store water with changes in soil organic carbon, particularly at a low silt or low clay content. Interestingly, we don't see the same responsiveness to soil organic carbon in calcareous soils. Out west, we may have some more experience with this, and we know that calcareous soils have divergent behaviors. And so I'm particularly excited to use this inherent feature to categorize soils for this and maybe other calculations, but for this calculation of the soil's capacity to store and deliver water. I know I showed a lot of data here. Here are the main points. Carbon and nitrogen indicators are closely related. Carbon and nitrogen indicators are responsive to organic amendments. Physical indicator measurement methods matter. 16S community structure drivers of climate and pH were pretty strong, but then within a site, we can see that manure changes the 16S community structure. Nitrogen mineralization may be well calculated using total nitrogen and carbon dioxide respiration, and available water holding capacity using intact methods is sensitive to changes in soil organic carbon, but not in calcareous soils. So I can't thank our partnering scientists enough. Without their careful curation of these long-term sites and their help with sampling and metadata accumulation, this analysis would not be possible. And then I also need to acknowledge the generous financial support of the Samuel Roberts Noble Foundation, General Mills, and the Foundation for Food and Agriculture, who saw the importance of this project with this giant scope and scale.
All right. That was a really great presentation. Does anyone have any questions that they'd like to ask Shannon? You can go ahead and use them on the in the chat box. I actually have a quick question. When do you think, I mean, you guys have collected a ton of data. Is there a timeline for publications or kind of getting this information out there?
Yeah, absolutely. Our first publication is out there. It's in the Agronomy Journal and it's open source, so anybody can find it. And that one is just introducing the North American project to evaluate soil health measurements. So if you Google Norris et al. 2020 and NAPSHM, which is how we abbreviate the North American project to evaluate soil health measurements, you can find that first publication. We are working hard right now to get our other publications ready. We have kind of a somewhat long peer review process because we have about 89 partnering scientists on this. And so we want to be able to provide them the drafts and make sure that they are comfortable with what we are saying before it goes out. And so we are in the, actually this week we have a meeting with partnering scientists to discuss our results, get some feedback with them in a Zoom type setting, and then papers will be going out to them in this review period. The goal, we have 9 other publications that we're working on right now. Ones that are specific to carbon, to nitrogen, to aggregate stability, and general framework for how we're working on these quantifications or quantification estimates of these functions. And so we have a lot of different balls up in the air, and they are all getting to the point where they are getting pretty close to send out. And so I would hope, by the middle of next year, we have a good body of literature on this project out there.
Great. I noticed that someone asked if the link to that publication could be posted. And then there's also the question of what are the most important biological indicators in soil health? If you can have a 30-second answer.
Sure. So often we categorize indicators into biology, chemistry, and physics. And I think one of the really interesting things about soil health is that, all of the work that we do in soil health is bringing to the forefront that biology is driving a ton of the system, right? So there's not just one thing that is a biological indicator. Aggregate stability is this combination of physical and chemical and biological factors that are all creating, whether or not you have stable aggregates. If I had to say one that was the most significant biological, I would probably say carbon dioxide respiration. It is the metabolic byproduct of aerobic respiration in the soil. And because there are so many microbes that are doing the work, that is one of the most influential of the factors that we have found, but we don't have our final recommendations at this point. So I would say that is my opinion on it.
Great. Well, thank you very much. Hopefully, if anyone else has any follow-up questions, you can always throw them in the chat box, and maybe Shannon can stick around and answer them.
Yeah, and I will post that link as well.
Great. We'll keep rolling to the next video.
Today's presentation is entitled Cost and Benefits of Nutrient Management in Soil Health Systems. The subject for discussion is nutrient management as a component of soil health. 3 topics of discussion are, number 1, to define soil health management systems. Number 2, define the procedure to determine cost and benefits. Number 3, present empirical examples from farm data. First of all, let's present the types of soil health management systems that we will consider. Number 1 is no-till. Secondly, reduced tillage. Reduced tillage could be reduced depth of soil disturbance, reduced surface area of soil disturbance, or reduced frequency of soil disturbance. This reduced frequency could be within a year or across years. Another type of soil health management system is cover crops. Generally, cover crops grow between 2 years of cash crops. Potentially, cover crops are grazed before planting the cash crop, or potentially cover crops are cut for forage before planting the cash crop. Another type of soil health management system is crop production with either rotational pasture or hay, that is either with or without grazing. An example of this would be having several years of alfalfa hay between cash crops reduces tillage across years. There's numerous benefits of soil health management systems. One benefit is increased soil carbon and organic matter, improved soil structure, decreased erosion, improved water infiltration, enhanced soil biological activity, and improved public water quality. A separate question is there a business case for soil health management systems? Specifically, do soil health management systems increase farm profitability? To investigate this, we'll use methods of cost and benefits analysis. The business case for soil health management system is determined by cost and benefits analysis. Adopting a soil health management system potentially reduces production cost. An example is eliminating tillage operations reduces equipment, fuel, and labor. Adopting soil health management systems potentially increases production costs. For example, planting cover crops increases seed and application expenses. Adopting soil health management systems potentially impacts crop revenue, either from yield increases or decreases that would be a benefit or a cost. So the question is, are the benefits of adopting a soil health management system greater than the cost? This is an outline of how the comparison is done. In the first column, or the middle column of the dollar values, we have the benefits. If you're in a conventional tillage system and you go to no-till, you have reduced expenses. You no longer have the equipment costs, the fuel, or the labor. So this is a benefit, reduced expenses. Likewise, after you implement the new system, you could have additional revenue. If the yield goes up, you have increased revenue. Comparatively, on the other side of the ledger, in the far right-hand column, we have cost. When you implement a soil health management system, you potentially take on additional expenses. For example, if you plant cover crops, you have the seed cost and the expense of planting the cover crop. If you have decrease in yield, you would also have reduced revenue. So when you sum both of the columns up, benefits and cost, you take the difference, the total benefits minus the total cost, and this is the change in the net farm income. Now let's look at the data and information that we'll be using for our analysis. During the previous year, the Soil Health Institute has conducted 125 farmer interviews. The first project is with the NRCS, Natural Resource Conservation Service. We have 25 interviews that were conducted in collaboration with the National Association of Conservation Districts. These interviews cover all the U.S. geographical regions with typical regional crops. Our second project is with Cargill. In this project, we conducted 100 interviews in 9 states with a focus on corn and soybean. And this are a listing of the 9 states. The information that we'll use as data are collected from farmer-reported observations. Our information is not from field experiments or replicated trials. Our focus is the cost and benefits of nutrient management. This will include expense comparisons of soil health management system nutrient alternatives. It will exclude changes in revenue. that would either come from yield, grazing, or forage impacts, which are not included. This is our analysis is focusing on nutrient management, and we just look at expense comparisons. The data for analysis are farmer observations for nutrients and rates of application. Many farmer interviews reported reductions in N, K, or lime. And these are the terms that farmers state to describe the soil health management system benefits of nutrient management: nutrient mineralization, nutrient recycling, increased organic matter, nitrogen credits from lagoon cover crops. And the economics is done with partial budget analysis, which determines the change in net farm income due to nutrient management. Without going into details of how this was done, I will present one reference that has all the information required to understand how we went about the analysis. This is an example of the way the data are collected from the farmers as we conduct the interviews. This will be an example for corn, a farmer going from conventional tillage to no-till with cover crops. When the farm ceased conventional tillage, these were the omitted practices. These are the expense reductions. The farm ceased chisel plow, 2 field trips with a field cultivator. The farm was using a typical row crop planter to plant the corn. Also, the farm was able to reduce 50 units of nitrogen with anhydrous ammonia. Also was able to reduce P by 30 pounds per acre and K by 20 pounds per acre. When abandoning and reducing these activities, the farm began in the no-till production, use these cover crop seeds. This was the cost per acre, $19 per acre for the seed. The cover crops were planted with an air seeder. And this farm example planted green, in other words, planting into the living cover crops with a no-till planter. The weed management, herbicide applications in these systems were the same. There was no change. These are empirical results, 12 farms from our NRCS data, average over 12 farms. These were aggregate. So they were all no-till, some were with cover crops and some were without cover crops. Looking at corn, we have the benefits, reduced expenses, some up to $70. Additional expenses, some up to $44.52. We have the same comparative numbers for soybeans. Let's focus in on the nutrient component of this. The farm, the average farm was able to reduce fertilizers and amendments by $34.34. At the same time, there was an increase in $3.29. This could occur for several reasons. Sometimes when the farms adopt soil health management system, they change. They may reduce nitrogen in one form, but they may come and use nitrogen in a different form. Will be one example. The other example, if they're planting cover crops and they're going to use it for either hay or forage, they sometimes apply nitrogen to the cover crop. So we see that for corn, the fertilizer was in amendments reduced $34.34. Comparatively, for soybeans, the reduction was $16.91. For these average farms, we see that the reduction in fertilizer and amendments is the largest category of expense reductions for corn and soybeans. However, we do notice that for corn, it's much greater. And this is because of the nitrogen applications in corn. These are results from a 22 farm average, Nebraska and Iowa, from our Cargill study. We do see similar results. For corn, the largest category of reduced nutrient reduced expenses for fertilizer and amendments. But coming over and looking at soybeans, we see that in these farms, the average reduction is only $1.42. This is because soybeans, the only potential reduction is P and K. Nitrogen is not used. Looking at both of them together, we see that the net change in income is about the same in each one, about $25 for corn in the Cargill collection, and then $9 for soybean. We say it's about the same thing for our 12 farm average from the NRCS data, even though the components of the expense reductions are very similar, or are not as similar for fertilizer and amendments. The totals do come out to be about the same. Now we'll take a look at summary and topics for further discussion from our empirical results. Soil health management systems entail nutrient management as a component of increasing farm profitability by enhancing soil health. Partial budget analysis itemizes field activities and inputs for comparing alternatives of nutrient management. Production practices that improve soil health and regenerate the soil, reduce expenses, and increase farm profitability. And then finally, agricultural production with the goal to achieve soil health as a component of economic efficiency.
All right, that was another great presentation. Are there any questions for Archie? Well, I can get things rolling. I never mind asking questions. Do you have any plans to do any of these economic budgets for any of the Western farms?
They're all the Cargill, of course, are in the Corn Belt area, but the NRCS study does have from all over the U.S. I just presented the ones that were corn and soybeans, and they were not in the far west just because they had similarity in the Cargill data. But when the full NRCS report comes out, there will be some Western states included in that.
Great. Do you know when that report will come out?
It hasn't been determined exactly. It's in the process. It should be within 2 or 3 months. It's in the process of being put together. Everything has been finished, but we have to have it formatted in the final form. That's the stage it's in now. It will be at the Soil Health Institute website with links so everybody can view it.
Okay. That sounds great. Does anyone else have any questions? No need to be shy.
Archie, will there also be some of these partial budget analyses done on other sites from our project where we have quite a few out in the West?
Yes, we're currently conducting that. It's not as far along as the NRCS and the Cargill data, but we're analyzing that. We'll have the same type results from using the farm management data of the sites that you described.
Any other questions from anyone? I guess I have another one if we have a couple minutes and to stay on time. Was there anything surprising with these partial budgets that you weren't necessarily expecting to see, but you saw?
From the NRCS data, no, I really can't say anything was not anticipated. As I described in the Cargill data, it was for these two states we've done so far. It was surprising that the fertilizer and amendment part was such a small decline, only about $1.50. But we have more to go. That was 22 states from 22 farms from those two states, and we have 100 in total. So we'll see what it looks like when we get everything done.
Great. Well, thank you again. If anyone thinks of anything, feel free to throw it in the chat box, and maybe you could include your contact information, Archie, if you don't mind, in case anyone has any burning questions that they'd like to follow up with you about.
Okay.
And I guess we'll just keep rolling. Thank you so much.
Thank you.
Good morning, everyone. I am Karen Hills. I am a research associate with the Center for Sustaining Agriculture and Natural Resources at Washington State University. I'm based over in Skagit County, and I am here today to talk to you about an ongoing effort currently underway to create a soil health roadmap for Washington State. Though I'm giving the presentation today, I did want to acknowledge the folks listed here on the screen who are really doing a lot of the heavy lifting in terms of this effort, and which is funded through the Washington Soil Health Initiative. So before we get into what's happening here in Washington, I wanted to give a little bit of a broader perspective on interest in soil health. So this is a map from December of 2019 showing different states in the US where healthy soils legislation has passed or was at some other stage in the process of passing. So this is, the point of this is just to say that Washington is not the only place where this type of effort is occurring. And it's exciting to see, such a broad group of states actually paying some attention to soil health. That, of course, interest in soil health is not new in Washington State. There's been work occurring over years and even decades that has led us to the point that we're at today. In 2019, proviso funding for the Soil Health Initiative passed and allowed us to take up a subset of activities smaller than the number of activities originally proposed, but I'm going to talk about those specific activities underway in just a minute. In terms of an update, in 2020, additional funding for the Soil Health Initiative passed both the state legislature and the state house, but unfortunately fell victim to the budget woes brought about by COVID, and so was vetoed by the governor. Our work under the proviso funding is still underway, and we're using the $250,000 funding to set the stage, hopefully for future efforts in this area. This slide shows the 4 major efforts currently underway. I am going to talk mostly about the soil health roadmap, but before I get into that, I'm going to touch briefly on these other 3 efforts that are happening. So the first project is the long-term agricultural research experiment. The proviso funding required that WSU establish this long-term agricultural research experiment, or LTARE, at WSU Mount Vernon Research and Extension Center. The ground is being planted to wheat this fall. Soil sampling is occurring to establish sort of baseline information, which will help with blocking the experiment. And then next spring, the different treatments will be initiated. And the size and the complexity of those treatments is going to be dependent on whether further funding is secured. The next effort I wanted to talk about is this Quick Fire Grants program. So the idea here was to get some money towards some outcome-focused projects. These are the 3 projects that were funded in late 2019 and are at sort of varying stages of completion. There's more information on these projects at the soilhealth.wsu.edu website that you can, keep tabs on what's happening with them. So critical to making improvements in soil health is understanding the status of soil health in a number of cropping systems. So the statewide soil health baseline assessment is a collaboration between WSU, with the WSU side led by Deirdre Griffin-LaHue, and WSDA led by Leslie Michael. So we're focusing on onions, potatoes, sweet corn, wine grapes, hops, tree fruit, and pulses to try to understand the current status, develop scoring curves and threshold values for soil health measurements. So if you get a soil health test result, you can understand what that means and how it compares to other fields growing a similar crop. And you can begin to evaluate how soil management practices are influencing these metrics. So over here on the left, you can see one of Deirdre's graduate students involved in the sampling effort. They sampled 97 fields in central Washington over the summer and have sampled another 34 potato fields in western Washington. So work on that is underway. So the 4th effort underway is the statewide soil health roadmap. And the goals of this effort are twofold. First, we wanted to document the current health situation, including goals and barriers to adoption of soil health practices. And then, as the name implies, this is a roadmap. So we're not only discussing the current situation, but talking about the way forward. So we wanted to outline a process by which public and private entities could coordinate efforts in reaching some of these goals and milestones. Now, as you know, Washington has a very diverse agricultural systems, and soil health, of course, in tree fruit may look very different than it does in, say, wheat systems or potato systems. So part of the challenge of this project is actually capturing that diversity. So we've handled this by identifying major cropping systems to focus on for this effort, and also identifying a WSU person who is already working within those cropping systems to lead the groups. So some of these folks have already been working in the soil health space within their respective cropping systems, or if they haven't, they generally have established relationships with growers and industry folks that kind of help get to that information in a timely way. So these are the cropping system groups and the folks that are leading them. You'll probably see some familiar names here with Andy McGuire in the Columbia Basin irrigated systems, which includes potatoes, Rich Koenig, the wheat and dryland systems, Gwen Hoheisel and Michelle Moyer are the leads on the grape and wine systems, Tiana DuPont on tree fruit, Deirdre and Gabe on Northwestern Washington Annual, and Doug Collins on the Western Washington Diversified Organic Group. And you'll see that one of these is not a cropping system. This brown square represents the environmental constituency. We wanted to hear from public and nonprofit entities who are interested in soil health from the environmental perspective. So Chad Kruger is leading the effort there. So this slide shows the process of creating the roadmap. We've identified the information that we want to gather and the key stakeholders who we want to gather that information from. And sort of the meat of this work is here, gathering input from key stakeholders. So to collect this information, we provided each group lead with a template of kind kind of questions, and then each lead determined the best way to go about collecting that information. So here's a couple of examples to give you the flavor of what some of this looked like. Over on the left, in December of last year, Deirdre and Gabe LaHue held a listening session in Mount Vernon. This was focused on the Northwestern Washington annual production. Over on the right is a word cloud that was created based on the responses that some of the participants provided for Doug Collins' Western Washington Organic and Diversified Group. And we used this to kind of zero in on the topics to discuss in our virtual sessions, which were held in March. So where are we now? Right now we're at the stage of extracting the key ideas and themes from all of this information that's been collected and compiling it into a single document. We will be seeking feedback and review from our advisory group and from external partners, and expect to have this document finalized and provided to lawmakers in 2021. This document will hopefully provide a lot of useful information going forward to inform activities at the LTAIR, resource allocation and policy development, as well as research. So we'd like to encourage you to stay engaged with the soil health work at WSU in whatever way you like to do that type of thing. The website at the top is soilhealth.wsu.edu, has kind of become a clearinghouse for this information on soil health work at WSU. There's also Instagram and Twitter accounts that you can follow. And also wanted to let you know that we have a SoilCon Washington Soil Health Week that's going to be happening February 8th through 12th next year. We have a website and there will be an agenda posted soon. So my email is at the bottom of the screen, as is the email of Chris Benedict, who is coordinating a lot of this work. Feel free to reach out with any questions that come up. And yeah, we'd love to hear from you, and happy to open it up for questions now. Thanks.
And just another great presentation. We're fortunate to have both Karen Hills and Chad Kruger with us. If you have any questions, follow up. I never mind kicking this off. I know you guys are sort of early in the process in terms of like the overall whole project, but do you have any, I guess, lessons learned if there's some states that are wanting to kind of follow in Washington's footsteps in terms of getting one of these roadmaps and doing this type of work?
Yeah, I can share a few thoughts on that, and Chad can chime in as well as if he'd like. I mean, I think the key so far, and the approach that we took was to work with folks that had existing relationships with growers and industry, and I think that has been critical to get the level of information that we were seeking. If it was just me starting out approaching all these folks that don't necessarily know me, I think we would have, the quality and the amount of information that we would have been able to collect was not as robust as working with extension and folks who already kind of have ins in these industries. The other piece was just from somebody who has done research. I started the process thinking, there was some importance to have like parallel approaches in each cropping system group, where for this type of work, we really left it up to the individual who is leading the process. Some cropping systems, there's already a lot of existing documentation on soil health, and others there's not. So, the approach that was taken kind of depended on what was existing in each system.
Yeah, I would add just one thing, and that's, we didn't get this idea, we didn't cook it up ourselves. So we kind of stole it from the Washington tree fruit industry, who has basically formulated their entire research and development game plan over the last 20 years around a roadmap for what the future should look like. And so I think that was really the driving idea here is, we all can have great ideas about what to do for research and directions to go. But when you engage the end users of that research and really try to define where do we want to be in 10 years or 20 years or 30 years, that really helps to solidify what's the priorities for the work you do.
Can I ask, we have a minute for another question?
Yeah, please.
Regarding the statewide assessment and the potential for developing regionally based scoring systems, have you done anything to coordinate with what we've been doing in Oregon? There was a NRCS funded project where we went around and sampled all over the state and have about 300 or so samples that we have used to make a preliminary scoring system for soil health in Oregon. But with the similarities between Oregon and Washington, I think if we wanted to increase that, we might be able to coordinate a regional scoring system.
Yeah, that's a good question. I'm gonna defer that one to Leslie, Michael, and Deirdre Griffin-LaHue, who are on this in this meeting. Deirdre, it looks like you unmuted, so you can answer.
Sure. Yeah, I can answer that. So we have been, I've been speaking particularly with Marcus Kleber and Dave Myrold, who I know did a lot of work in potatoes in Oregon, Washington, and Idaho. And I've spoken with Dr. Myrold also about the NRCS project. So we are discussing ways to kind of build a more regional database that includes their data and the data that will be coming from our project.
Yeah, so that potato project is a separate one. Dave has another NRCS project, and then there is a third one where we have, these 400 samples that was in order to make a scoring system. And so it has a very diverse kind of cropping systems with orchards and with vegetables and with grain crops as well. So be happy to talk more about that at some point.
Yeah, that would be perfect. Thanks.
It looks like we have another question from Ben Harris. Are there opportunities to get involved from a specific crop angle? He works with the U.S. Potato Board and thinks that the Potato Sustainability Alliance would also be interested.
Yeah, thanks for the question. This is probably a great stage to ask that question at. We are working with the Washington Potato Commission, and we would definitely like to chat more with you and the U.S. Potato Board and Potato Sustainability Alliance, kind of as their work pertains to potato production in Washington. So definitely shoot me an email. My contact info is at the end, and I'll also put it in the chat box.
And then we also had someone who indicated, gave their contact information. So just wanted to point you in that direction. It doesn't seem like they asked a question per se, but we have a couple more minutes if anyone has any follow-up questions.
I'll just weigh in on the last question and Gary's comment that, this initiative is a multi-institution agency partnership with a lot of support from industry. And so we didn't just cook this up out of thin air. It really came from a lot of groundwork, a lot of partnership and discussions that go back years, in fact, almost a decade. And the opportunity came up. So WSDA and the State Conservation Commission are also the other two agency partners. And of course, the Conservation Commission connects with the conservation districts and then also the industry. And potatoes has been the #1 supporter from the industry side.
Yeah, hello, this is Gary Barr with the Washington State Department of Agriculture. And Karen, yeah, thanks for your talk, and Chad for those comments. So we're really glad to be working with the WSU and Conservation Commission partnership on the Soil Health Initiative, and we're looking forward to having work hopefully fully funded. We did get a little funding here this last legislative session with the bill, when the bill got passed. So the Department of Ag does have some funding. We are under a hiring freeze right now, though, unfortunately, so we're not able to hire a lead person in Olympia quite yet for the Soil Health Initiative, but we hopefully will get that underway when the exemption gets lifted, hopefully. So thank you.
Thanks,
Yeah.
Gary.
Thank you again for your great presentation, and we'll keep moving. We've got two more presentations, and then we'll take a break. So hold tight, and yeah, we'll just keep going.
Hello, everyone. I'm happy to share with you some information on our 2019 sugar beet study focused on nitrogen and irrigation water use. The study is in collaboration with Dr. Sana Shafian. She's our precision agriculture specialist in Moscow, Idaho. And the work is supported by the Idaho Sugar Beet Growers, Snake River Sugar Beet Research and Seed Alliance, as well as the Amalgamated Sugar Company. Our main objectives were to analyze the effects of water and nitrogen fertilizer rates on sugar beet yield and quality, as well as to explore the potential of using aerial-based, UAV-based data for sugar beet nitrogen and water content monitoring. We also, in the study, wanted to assess the feasibility of predicting sugar beet root yield, as well as recoverable sugar using UAV-based sensors. And also, we conducted extension outreach focused on water and nutrient management in sugar beets, remote sensing, and using UAVs for crop monitoring. So to explain a little bit about the study, the trial was conducted at the University of Idaho Parma Research and Extension Center. We only used one variety because this is just an initial work, and we're hoping to include at least 2 or 3 varieties in our expanded study planned for next year. We used the typical planting date and row spacing and seed spacing. We had 4 rows in each plot. The plots were 40 foot long at planting. Then they were trimmed prior to harvest. We had 6 replications for each treatment. The previous crop was corn, and the plots were irrigated with subsurface drip irrigation system. Here are some of the pictures showing sugar beet planting, our irrigation system, and the collection of biomass samples in season. As an introduction, sugar beet production profitability is based on maximizing these 3 parameters: beet yield, sucrose content, and sucrose recovery efficiency. So efficient nitrogen and water management are key for successful sugar beet production in general. Nitrogen deficit in the soil can reduce root and sugar yield. So over-application of nitrogen can reduce sucrose content and increase nitrate impurities, which in turn lower sucrose recovery. Application of nitrogen in excess of sugar beet needs may lead to vigorous canopy growth, but it can compromise root development as well as sugar production. In our recent research in Idaho showed that about 60% of all evaluated sugar beet fields application of nitrogen did not increase sucrose yield. So this may suggest that residual soil nitrogen from past applications and the in-season nitrogen mineralization provided adequate nitrogen for sugar beet growth, and indicates that growers could maximize sugar yield and save money by applying less nitrogen. Appropriate irrigation amount and timing can optimize sugar beet yield, and it should be minimizing disease pressure, can lower water and energy costs, and it can also minimize nitrogen leaching. Excessive irrigation, on the other hand, can increase sugar beet root yield, but at the same time typically lowers the sugar content. And of course, the remote sensing is a very promising tool for in-season nitrogen and water management, as well as for in-season prediction of yield and quality, which in turn can improve the economic returns to sugar beet growers and processors. We had a very well attended crops, soils, and technology field day last June. We showcased this trial, and we explained the sensor-based part of this work, and we also had a drone demonstration. And unfortunately, this year our field day had to be canceled this summer. I wanted to also share with you that we publish our research updates on the webpage, the Idaho Crops and Soils blog, and this is part of our web-based extension outreach, as well as helps with accountability for the funding institutions. So far, we've had visitors from 106 countries and every single state in the United States. For our nitrogen treatments, we had 3 nitrogen rates: 100, 200, and 300 pounds of nitrogen per acre. Nitrogen was applied as urea at planting. And it is difficult to find fields that are very low in residual soil nitrogen. So we had 116 pounds of nitrogen per acre pre-plant. So therefore, our 100 pounds per acre rate was actually 116. For our water treatments, we used a water scheduler developed by a team of irrigation engineers from the University of Idaho and the Washington State University. And this schedule is based on the evapotranspiration values obtained daily using the AgriMet weather station located in Parma, Idaho. So the daily reference grass-based ET was then adjusted using the sugar beet crop coefficients to make the irrigation decisions. And we imposed 2 water levels, 100% and 50%. And the irrigation water was applied using the subsurface drip irrigation system, and the tape was buried down to 7 inches. We utilized the Matrix 100, the unmanned aerial vehicle, or UAS, or drone, which was equipped with the Red Edge camera. And the camera has a capability to collect spectral reflectance in blue, green, red, near-infrared, and red edge wavelengths. And the drone data were analyzed using the MicaSense Atlas software, and then the Pix4D image analysis software. And today I'd like to share some of the results for the biomass, sugar beet root yield, and ERS, which is the estimated recoverable sugar data. This image shows the plant stand differences between the low water, low nitrogen treatment, which is on the left, versus the high water, high nitrogen treatment on the right. This is a UAV derived image, and the orthomosaic UAV image is basically composed of multiple images which are stitched together, and it is properly scaled and geographically correct. The relationship between reflected, absorbed, and transmitted energy is used to determine spectral signatures of plants. And this is illustrated on the left image. The right image shows how the normalized difference vegetation index, or NDVI, compares the reflectance values of red and near-infrared regions of electromagnetic spectrum between healthy vegetation and stress vegetation, where healthy vegetation is typically associated with a higher absorption of red and reflectance of near-infrared light. And so you can see in this case we have stressed sugar beets and that have higher reflectance in the visible region, and on the other hand there is a reversed pattern for stressed sugar beets in the non-visible range. Here's what we've seen in terms of the UAV spectral indices, NDVI specifically in this case, and the sugar beet biomass. The graph on the left shows the relationship between sugar beet plant height and NDVI measured in June, and then the figure on the right shows the relationship of the sugar beet biomass dry weight and NDVI also measured in June. And previous work has shown that plant height and biomass weight are plant characteristics directly related to light interception and as well as spectral reflectance. So in our trial, a strong positive linear correlation of NDVI with sugar beet plant height and biomass weight were observed in 2019. For the UAV spectral indices and the sugar beet leaf nitrogen content, previous work showed that sensor readings were strongly related to the sugar beet total nitrogen content at all sites and nitrogen rates. And in our study, all indices were equally well correlated with sugar beet leaf nitrogen content. So we are showing here blue, green, red, near-infrared, NDVI, and red edge-based indices and their correlation with leaf nitrogen measured in June in this case, and similar data was collected in July as well, similar results. What we're also seeing is that as leaf nitrogen content increases, the reflectance in red region goes down and near-infrared goes up. For the sugar beet root yield results, the 200 pounds of nitrogen per acre in combination with 100% applied water maximized sugar beet root yield in our study, and then increasing nitrogen to 300 pounds of nitrogen per acre did not increase yield any further. And then also what we found is that 100% water significantly increased sugar beet yield compared to 50% applied water, which is not surprising. The next step is now to evaluate different water rates in the future. Then these graphs show the relationship between sugar beet root yield and NDVI values obtained early in the season in June, which is shown on the left, and later in season or in July, shown in the right figure. So the sugar beet yield estimation accuracy is increasing as we expect it to happen between early season to late season as the growing season progresses and sugar beet plants develop. And in our study, the yield was estimated with 65% accuracy using June data and the accuracy then goes up for the July data to 88%. Finally, for our estimated recoverable sugar, or ERS results, 200 pounds of nitrogen per acre in combination with 100% water maximized the ERS, and then increasing nitrogen further did not increase ERS. Just like with sugar beet root yield, the 100% water had significantly greater ERS compared to 50% water. And here's the relationship between the estimated recoverable sugar and NDVI. Again, the June data is on the left and the July data is on the right. And just as with the sugar beet root yield, the estimation accuracy goes up from 75 to over 90% between June to July. So our results agree with previous work by Boo et al. In 2015, where they found that using the handheld sensors at 12 to 14 leaves resulted in stronger relationship to sugar beet root yield and estimated recoverable sugar compared to readings collected at 6 to 8 leaves growth stage. To summarize our 2019 results, what we've seen was that 100% water treatment resulted in significantly greater yield and estimated recoverable sugar compared to 50% water. And the future work, we would like to evaluate whether we could cut down water amount by 20 or 25% at least. And then for sugar beet biomass, what we've seen is that all spectral indices we evaluated performed equally well, and all of the indices we looked at were excellent estimators of both plant height and leaf nitrogen content, and yield and estimated recoverable sugar should have similar trends as well. And then accuracy of yield and ERS prediction from UAV spectral indices improves substantially from June to July, so relationship becomes linear also later in season. And the overall conclusions from this initial work is that yes, UAV-based data can be successfully used to estimate sugar beet root yield and quality, in this case, estimated recoverable sugar in season. And this experiment was repeated in 2020, and we are analyzing the results.
Unfortunately, Olga was not able to join us this morning for a live Q&A, but I am putting her email address in the chat box in case anyone has any questions and wants to reach out to her. She said that she's more than welcome. If anyone wants to email her or give her a call, she's more than happy to answer any questions that you all may have. Is there any other questions burning from any of the previous presentations? We have about 3 minutes before the next presentation. Or any comments? I just think that this session is just super great, and there's so many awesome presentations, and I'm definitely learning a lot, even about just the stuff going on here in the West, let alone across the country. But yeah, it's super exciting. Well, we're only a couple minutes ahead, so I will probably queue up this next video because I don't want to just hang out for the sake of hanging out per se. Although continue if you've got questions to throw those in the chat box. Yeah. Hopefully I'm not stumbling as badly with all of this screen sharing. Okay, here we go.
My name is Ryan Boylan. I'm the Research and Monitoring Coordinator with the Palouse Conservation District. And I'm here to talk to you today about a soil health data communication education tool that we've developed for the Inland Pacific Northwest. I just also want to acknowledge the other folks that have helped contribute to this, which is Nick Harris and Zach Carter, who both work at the Palouse Conservation District, and Tabitha Brown, who's a colleague of ours, who works with Farmer in the region. The take-home message for our talk, or kind of the general theme, I guess, is that the Palouse Conservation District has been working to develop an interactive soil health data portal with the goal of creating a platform for producers to share data and learn from one another in their on-farm trials. Before I get into the presentation, I just want to give you a quick overview. I'll talk about the background of where the Palouse is and where we do our work. Talk very briefly about a big conservation effort that's happening, and then how that has led into us developing the soil health data portal. All right, with that, I'll show you a picture of the Palouse River watershed. This is the Palouse River watershed. It's located in eastern Washington and parts of northern Idaho. It's just over 2 million acres, and 67% of that is in agricultural land. In the heart of the Palouse River watershed, right in the center, is Whitman County. And as you drive into Whitman County, what you see is a road sign like this that says the nation's leading wheat producing county. Whitman County and the Palouse in general has some of the highest wheat yields in our country, which means lots of agriculture. All the water from the Palouse River watershed drains over Palouse Falls, which is Washington State waterfall, into the Snake River. And the Snake River is critically endangered habitat for fall and spring Chinook salmon. So all the work that we do up here goes downstream and helps to protect salmon as well. As I mentioned, the Palouse has some of the nation's highest wheat yields, and all that comes down to the soil. So we have these really deep wind-blown loess soils that have been generating high wheat yields for a long time. Starting in the 1890s, folks came and started farming the land, and you can kind of see the progression here from horse-drawn equipment. In the early years and like up to the 1970s, there was a lot of issues with soil erosion. And they estimated that 3/4 of a ton of soil was lost for each bushel of wheat that was produced. And annual average soil erosion rates range from 10 to 80 tons per acre per year. Since the 1970s, the advances in farming practices and technologies such as no-till drills and direct seeding and the implementation of conservation practices have reduced these erosion rates to 5 tons per acre per year, which is great. We've almost cut that in half, or by 400% in some cases. But if you think about 5 tons per acre per year and roughly 1.7 million acres that are in agriculture, that's still a whole heck of a lot of erosion that we see entering our water bodies. So in 2015, this group of 19 partners came together and we wrote a grant to the Natural Resource Conservation Service, and we received it, and the Palouse River Watershed Regional Conservation Partnership Program was formed. The whole goal of this program was to improve water quality, soil health, and wildlife habitat in the Palouse River watershed. And the main objectives were to install a whole lot of conservation practices, including 45,000 acres of conservation tillage, 450 acres of riparian buffers, and 530 acres of conservation easements. Since 2015, we've accomplished all of these goals, which is great. So we pat ourselves in the back for that. And then we've also got an extension. So over the next 5 years, we will be implementing just as many of these. The reason I talk about the program and the big effort, the partnership that we formed, is because that's enabled us to work with a lot of producers in the area. And I was hired in the start of that grant to set up our research and monitoring program. And one of the sections of my program is the soil health program. And so I work with a lot of farmers all over the Palouse River watershed. And our goal initially was to identify regionally specific trends and soil health indicators over a range of management practices. And some things that have kind of come out that since that original goal was stated is a lot of the work that we do now is to support on-farm research trials for folks that want to try things. One of my favorite parts of my job is when somebody comes to me with an idea and they ask if I can help them assess that idea, or at least soil sample out there to see if it's making a difference. And then what I'm going to talk about today is facilitating peer-to-peer learning through a soil health data portal. And I just want to touch briefly on the management practices that we're addressing or that I'm talking about. I should have said this in the beginning, but all of the agriculture here is dryland ag. And historically, there was a lot of tillage, which led to all that erosion. So we're still assessing some of the conventional tillage. There's mulch till or conservation till practices that we're assessing. True no-till practices, no-till practices that have started to integrate cover crops. Then there's interseeded cover crops with cash crops. There's folks are starting to integrate livestock and grazing those cover crops. We're assessing those. We also have a couple, or a restored prairie site, a native prairie site. We've sampled on CRP fields and then also in a couple of riparian areas. Here's just a quick map of our study locations. The gradient in colors there just indicate the farming zone. So as you move from west to east on the Palouse, you go from a grain fallow sort of system into an annual cropping system. And the typical crop rotations in the annual cropping system would be winter wheat, followed by a spring grain, followed by a legume. So within our study, we have 17 farms and 54 fields that we've assessed since 2016. We collect samples in the fall and the spring. Originally, we were sampling at 4 depths, 0 to 6 inches, 6 to 12, 12 to 24, 24 to 36. But currently, we've scaled back on that and are just doing 0 to 6 and 6 to 12, the two. It saves money and time for all of us that are out there, and we're assessing a lot of fields. And so within each field, we randomly select sampling locations which are representative of the topography and then also of the different soil types that are out there. So for each field, we composite the samples, and there could be up to 15 stabs taken per field, and then those are put together to make one sample and sent to the lab. For the data analysis, we're essentially sending these samples to 3 separate labs. The first one is the Midwest Laboratories, and they're just kind of doing our traditional fertility tests. The second one is Woods End Lab, and they're doing kind of our soil health overview. And then the third one is Earthfort, and they're doing soil biology for us. So all this data is stored on our PCD server, and then each year we share it with participating farmers. And so now I'll just jump into showing you what the soil health data portal is. But before I do that, I just want to say, one of the reasons that this was created was I was spending a lot of time each spring and fall just packaging all the data that we were getting back from the labs and sending them to people or to the participating farmers. And one of my coworkers, Nick Harris, who was mentioned in the beginning, we had the idea of putting together a little application that could display the data for people, because getting 7 to 10 PDFs in the mail or via email can be sort of hard to synthesize. So we're like, oh, we'll just do this. It'll save us time because we can just put the data into this application and then they can view it. And then they can also download it from there. So one of the reasons this happened was maybe because we were getting lazy or trying to be more efficient. So yeah, this is what the soil health data portal looks like. This is just the landing page. we have a little video tutorial about how to work the tool itself. And then each producer can click on that blue box where it says click here to get started, and they all get a producer ID that's emailed to them. They fill in that producer, they type in the producer ID and then click proceed to view your data. And this is kind of what it looks like once they get in there. You can see on the left-hand side here where it says data inputs, producers can select their specific field. It depends on the person, but there could be up to 5 fields that we're sampling for these folks. They can select the type of analysis, and we've broken that down by biological, chemical, soil health, and physical. And then they can select their data grouping, desired parameter, and the test. And this is just kind of a close-up of the figures that pop up when they go in there. There's essentially 4 things going on here, and I know it seems like a lot, but I'll just walk through them really quick. So there's a map in that top left-hand corner of the fields that we've been sampling. That top figure here just shows seasonal averages by depth of the specific parameters. So for this instance, it's organic matter. And you can see it's color-coded by year and season and by depth here. The second figure is a management comparison. Oops, sorry about that, is a management comparison. And these are just box plots of all the different management practices that we've been sampling. And you can see those red circles there are indicative of the management practices that this particular producer has on their farm, has on his farm. So you can kind of see how the samples that we're collecting on their place stacks up against or across multiple management systems or management practices. And then the final, I'm just gonna move my video real quick. The final figure that we have here is a regional comparison. So this is a concentration or percentage. In this case, it should be percentage organic matter by depth. And then each one of these different colored bars represents a farm average here in green, a field average, so for this particular field, RBFB that we've selected, and a regional average. And this is all based on the management practice that they've indicated to us that they're using in that field. Another thing we've been working on, because we can just, for this particular application, you can just show one field. We have this field comparison. So an individual could select to look at organic matter across all their fields, and that's what this plot shows. Is for the 6 fields that we've sampled out there, they can just see how that's changed over time and by field. So in this case, you can see that RBFD and RBFE have lower organic matter concentrations than the other 4 fields that we sample. Yeah, and this is just another tab that's at the top of that landing page once they enter their ID. So yeah, that's a brief overview. We've really just gotten started doing this. And the next steps would just be to continue to develop the tool. There's still some bugs that we're working out. And you could probably see some red text in that previous slide. And that's just a kink that we're still trying to work through. We're going to continue to work with producers to make the portal more user-friendly. I've presented this to 3 of the folks that we work with. And the very first iteration of this, they were like, I don't know how to use this. You have to walk me through it. So that kind of prompted us to have a video, and then I've walked them through it, and some of them have found it useful. We also want to hold a workshop with several of the producers that we're working with to see what we can all learn from the data that we're collecting. So have maybe sometime in the first half of that day just to show the tool and get people familiar with how to use it, and then get folks talking about how this information is useful for them and then what they've been learning from the trials that they've been running on their farms. And then the final thing that we're going to be working on is, we've been collecting data for 3.5 years now and analyzing that data to try and achieve that initial goal of identifying regional trends and soil health indicators. And with that, I would be really happy to take any questions. I have my contact information down in the bottom right-hand corner there, and then Nick Harris, who's also worked on the application with me. I should also probably thank the folks that support all this work. It's the Washington State Department of Ecology, Washington State Conservation Commission, and the Natural Resource Conservation Service. And with that, we'll go offline and answer any questions.
Great. Ryan is with us this morning for a live Q&A. I see that there's already a question in the chat box, which is great. So feel free to type in some more questions, but I'll ask Ryan this one. This website database, is this website database for producers, or is it a custom-made site? And then they go on to ask, is this a program or a plugin? And could others easily replicate this to share data with their clients?
Yeah, that's a really good question. So the website and database, we custom made essentially. So it's all been programmed in R using Plotly and Shiny. So it's kind of housed through a Shiny server, and then we put it out there. In theory, it could be easily replicated though. The code's already written, and you would just have to have the data in a similar format to the way that we put it in, and it could be expanded out probably.
I actually have a follow-up question for you. Is this site password protected? Can anyone look at anyone's data, I guess?
No, yeah, that's why we gave each producer an ID. So if you had their ID, in theory, you could go in and look at their data. But if you don't, it would be harder. I'm sure it could be maybe easily hacked into if somebody was really eager to get some soil health data. But we try and protect it because it is their information, and that's something that we work with all the time, is protecting personal information for the landowners that we work with. So yeah, that producer ID kind of protects us against that.
And I also have another follow-up question.
Yeah,
are you providing any sort of, messaging, or, so producers are going on here, they're looking at their data and maybe their fields are ranking low, or I noticed on that organic matter graph, there was a really big spike in percent there at the end. And whether or not when you're quality checking this data and you're like, oh, this is probably what's happening, if you're using any messaging to explain, or also to talk about, oh, your organic matter is low, so here's some things that you can do to increase it, or anything like that.
Yeah, so that's a really good question. I always feel like we're kind of in a funny spot. I don't tend to give recommendations, like on-field recommendations, because I'm not like a certified agronomist or anything like that. It's fun to speculate with the farmers what's happening, or why you would see like a 1% increase in organic matter, and that could just be an anomaly in the data. So that's like obviously something that we talk about when we send it to them. And so it's been really cool to just have these conversations. Like, it keeps the conversation going, and then the bouncing ideas off of each other. But I'm definitely not giving specific recommendations. Like, that's not my job. It's just to kind of collect the information, show them what we're finding, and then synthesizing it together. Great.
There's a question that just popped up. Do you have any plans to include or expand the comparison tool so producers can see how their fields compare to other fields across Palouse County, rather than just comparing their own fields?
Yeah, so it's still kind of a work in, like I said, it's still kind of a work in progress. We did write a pre-proposal for a Western SARE grant last spring to try and expand the tool. We were going to work with the American Farmland Trust. Unfortunately, that wasn't funded. So we're just kind of slowly creeping along with it now. We did try and get that comparison with other fields for that management comparison, the box plots I showed. And then there is another figure that has like the regional average versus their farm versus their field. So it sort of gets at that. As I mentioned, I think it'd be great to have the group in a room so we could all talk about what we're finding on their fields, and then they could have conversations with each other and not just with me. But yeah, it's still a work in progress. And I think I mentioned this in the presentation too, but it is a little overwhelming even for me to look at, to just have like three figures and a map thrown at you. So I think some of the next steps would be to simplify that and find and work with the producers that we work with to identify the parameters that they're most interested in, and then have those be the first several things that they land on and then move from there.
Great. Yeah, I mean, it's a really interesting tool and I'm excited. Hopefully you'll continue presenting about it at various conferences and modes, I guess. And yeah, just excited and interested to see how this progresses. So we are going to take a 20-minute break. So everyone can get up, you can walk around, you can get some coffee, some water, whatever it is you need to do. And we'll meet back here at 10:30 Pacific time, 11:30 Mountain time, and you can make those calculations in your head if you're not in either of those time zones. And we'll get rolling, and we have essentially 2 presentations left, and I'm pretty excited about them. So I hope you all, if you do step away, you come back in 20 minutes. And I'm gonna throw up the schedule just in case you hop on and off, just so it's an easy reminder of what we're doing. And in the meantime, again, as always, if you have any follow-up questions and want to throw them in the box, and if that presenter has already left, I can get contact info and get you guys connected. Okay, see you all in 20 minutes.
research physical scientist for the USDA in Kimberly, Idaho.
Welcome back, everyone. Hopefully you had a nice break and are ready to get back into this session. Like I said before the break, we have 2 presentations left. One is a standard 20-minute presentation, and then our last one is actually longer, 40 minutes, taking up 2 time slots. Originally, this was going to be one of the keynote talks, and there's a lot of really neat information, and so it definitely warranted a little bit longer. And so we'll be done in an hour. So if you will bear with us, I'll queue up this next video. And as always, if you have any issues with audio or visual, just put a message in the chat. If you have any questions for the presenters, I know that both of these presenters are going to be online. Feel free to throw those in the chat as we go along or at the end of their presentation. Great. Well, let's get going. Oops.
Hello, my name is Andrew Beer. I'm a research physical USDA in Kimberly, Idaho. This talk is centered around soil organic carbon dynamics in 3 of our research locations. First, I'll provide a little background on the 3 research locations. Then we will look at some of our observed data for soil organic carbon to answer some interesting questions about how management is affecting soil organic carbon levels. Don't be afraid, but we will also walk through some statistical analysis. One big part of what I've been working on recently is the use of a model to simulate each of our research locations. So we will have an introduction to models, discuss the model that I utilized, and talk about some of the problems I encountered while modeling our research locations. Finally, we will conclude by going over what the model projects will happen to soil organic carbon up until year 2050 in our 3 cropping systems. Our 3 research locations are affectionately referred to as the cover crop study, the GraceNet study, and the long-term manure study. The cover crop study was initiated in fall of 2015 on a previously furrow irrigated field. It's a continuous corn cropping system where we're testing 3 factors: tillage, manure, or winter cover. Plots were arranged with or without manure application, with or without winter cover in the form of triticale, and with conventional tillage or no-till practices. Here, manure was applied on a dry weight basis at a rate of 30 tons per acre. The GraceNet study was initiated in fall of 2012 on a linear irrigated field. This is a typical dairy forage rotation of corn, barley, and alfalfa with treatments of fall or spring applied manure, fall applied compost, and spring applied urea or super urea, as well as a no treatment control. Here, manure was applied at a rate of 30 tons per acre, and compost was applied at a rate of 15 tons per acre. The long-term manure study was initiated in fall of 2012 on a sprinkler irrigated field. It's a commercial cropping rotation of wheat, potato, barley, and sugar beet, and revolves around variable manure application rates and application frequencies. Annual or biennial manure application was paired with application rates of 8, 16, or 23 tons per acre, with the addition of a synthetic fertilizer treatment and a no treatment control. Okay, the first thing we will look at is the long-term manure study. We will follow with the GraceNet study and then talk about the cover crop study. The first thing we are looking at are the raw values of soil organic carbon we obtained for each treatment in each year. Here, table cells that contain the same letter within each column are not significantly different from each other, but we will walk through the most important points. On the left, you will see treatment designations, A for annual application and B for biennial applications. I'll add red arrows at the bottom of the screen to indicate years that received manure from biennial treatments. One overall trend we saw was that after harvesting either potato or sugar beet, the mixing of the soil that occurred during harvest probably reduced soil organic carbon test values compared to the preceding year. This has nothing to do with treatments, but you should consider this if you are tracking your soil organic carbon in a rotation that includes either crop. You may consider comparing soil organic carbon at a standard point in time in the rotation, like in the fall after wheat in our example. By 2019, there was no significant difference between the synthetic fertilizer and the no treatment control. By 2019, in all cases, the annual applications resulted in numerically higher soil organic carbon levels than their biennial counterparts. Lastly, as one would expect, as the application rate increased, so did the average soil organic carbon content. So then, how much is soil organic carbon changing each year on average? To assess the average change in soil organic carbon per year, simple linear regression was used to plot soil organic carbon level as a function of year among the study treatments. The resulting slope estimates and confidence intervals provide some inference. The estimate is our best estimate of average annual change. The confidence interval states that we are 95% certain that the true slope is between this interval. This analysis indicates that applying at least 8 tons per acre annually or 23 tons per acre biannually will result in building soil organic carbon. The estimated slopes indicate that you will probably build soil organic carbon under lower application rates. However, as the confidence interval for these treatments included 0, we cannot be certain. Moving on, a couple other questions we wanted to answer are, is there a distinct difference between annual and biennial application? And can we predict soil organic carbon change based on application frequency and application rate? To test if there is a difference between the annual and biennial manure applications, we used a linear mixed effects model and analysis of variance on a subset of data. Remember those statistics? Well, we're going to walk through some. First, the model indicated a significant effect of application frequency. However, it also indicated that the effective frequency is dependent on both the rate of application and which year we look at. To clear things up, we performed an additional contrast test between 2 specific treatments we found interesting, 8 tons per acre annually and 16 tons per acre biannually, as theoretically they add the same amount of carbon to the soil. Here there was evidence that they had different effects on soil organic carbon level in only 2013 and were otherwise equal. So there is a difference in annual and biennial manure applications, but that difference is dependent on the application rate and year of measure. In other words, there is evidence that an annual application at rate 1x is not different than a biennial application at rate 2x. However, considering other combinations of rates and frequencies complicates the problem. Now, let's finish discussion of the LT manure study with some simple predictive modeling. Here, the objective was to obtain a prediction equation for soil organic carbon levels or estimated change based on manure application rate, application frequency, and years in the system. This was achieved using multiple linear regression fitting rate, frequency, and years as continuous effects. Although the multiple linear regression method has flaws for this type of data, it allows us to make inferences on the average effective treatments that we did not necessarily test. The example that is shown is the modeled effect on soil organic carbon levels after 5 years of manure application at a rate of 5 and 10 tons per acre applied annually, a frequency of 1, biennially, a frequency of 0.5, and triennially, a frequency of 0.33. This is only a simple model based on our observed data, but if you would like to know what it says about your system, feel free to email me. Now discussing the GraceNet study, here are the raw values of soil organic carbon for each treatment in each year. Cells containing the same letter within each column were not statistically different. I've also placed red arrows beneath years where the manure was applied. At no point in time were soil organic carbon levels of either synthetic fertilizer treatment different from the control. By 2019, soil organic carbon under the fall manure application was numerically higher than the spring application, but this was not statistically significant. This particular analysis suggests that there isn't a difference between applying manure in the fall or spring when it comes to accumulating soil organic carbon. Similarly, by 2019, the average soil organic carbon under the fall compost application was numerically between the spring manure and control treatments, but not significantly different from either. So it is inconclusive what effect fall compost application had on soil organic carbon accumulation. Let's move on and see how much soil organic carbon is changing per year on average in this dairy forage rotation. Here, we are looking at simple linear regression analysis again. The confidence interval for the slope of both synthetic fertilizer treatments and the control included 0. Therefore, we cannot be certain that soil organic carbon changed at all in these treatments. The remaining treatments accumulated soil carbon with estimates in the descending order fall manure, spring manure, and fall compost. This analysis suggests a slight benefit to applying manure in the fall versus in the spring when considering soil organic carbon accumulation. Moving on, 2 questions we wanted to answer were, is there really a difference between fall and spring applied manure? And how did the 3 years of alfalfa affect soil organic carbon levels? To answer whether there really is a difference between fall and spring manure application, a linear mixed effects model was used in conjunction with analysis of variance on a subset of data. Here, fall or spring manure application is considered a timing effect. The results indicate a significant main effective year, but no significant main effective timing. The timing by year interaction could be considered marginally significant, meaning we can conduct further contrast testing to assess the effect of timing in various years. Here, there was strong evidence that there was no difference between the effects of timing on soil organic carbon levels from 2012 to 2018. In 2019, a significant difference was indicated. Upon further inspection, these results can be explained by the suspiciously large difference between fall and spring manure application in 2019. Practically, I find that significant evidence is lacking to conclusively state that fall manure application results in greater soil organic carbon accumulation. Now, how did the alfalfa affect soil organic carbon levels? To assess if alfalfa had an effect on soil organic carbon levels, data was subset for the first and last year alfalfa was grown, and a paired Welch's t-test conducted that assesses if the difference in means, in our case soil organic carbon levels before alfalfa and soil organic carbon levels after alfalfa, is different from 0. There was no evidence that the difference in means was different from 0. In other words, soil organic carbon did not change during the 3 years of alfalfa. That being said, let's move on to the cover crop study. Here are the raw values of soil organic carbon for each treatment in each year of the cover crop study. Cells containing the same letter within each column were not statistically different. First, the initial values of soil organic carbon were approximately 30% lower than at either the GraceNet or long-term manure studies. This is likely because the site is located at the head of a historically furrow-irrigated field and slowly had soil carbon erode over time. From 2017 to 2019, the only obvious differences in soil organic carbon were between treatments with manure application and those without. Let's see how much soil organic carbon is changing per year on average. This is another simple linear regression analysis. This time, the confidence interval for the slope of all treatments was above 0, indicating that all treatments observed soil organic carbon accumulations during the period of study. Treatments where manure was applied had considerably higher soil organic carbon slope estimates, while there was no obvious impact of winter cover or tillage practice. One thing I should note is that although soil organic carbon is seen here to accumulate quickly, these rates would not be expected to continue in perpetuity as soil organic carbon levels reach plateau levels based on soil properties, climate, and management. One bit of anecdotal evidence are the records of the Idaho Dairymen's Association, which has records from sites with a 20 to 30 year history of manure application, and most soil organic carbon levels are between 2 and 3%, with only a few over 4%. To see if the effects of tillage practice or triticale cover could be separated from the large effect of manure, a linear mixed effects model was used fitting the factors of manure, cover, tillage, and year. The analysis indicated significant main effects of manure and year. However, there was also significant interactions effects suggesting that the effect of manure varied by year, the effect of tillage varied by year, and the interaction of manure and tillage varied by year, which was further explained using contrast testing. The interaction between manure and year could be explained as the first year soil organic carbon was measured, manure had not yet been applied. The interaction between tillage and year indicated there may be a marginal difference in how tillage practice affects soil organic carbon levels in 2019 only. The three-way interaction clearly indicates that tillage practice affected soil organic carbon levels only in 2019 and only where manure was applied. Practically, I believe this was due to the very high soil organic carbon levels obtained under the no-till, no-manure triticale treatment in 2019, and there is not significant evidence to suggest that tillage practice has actually affected soil organic carbon accumulation in the system. But keep in mind, these are small research plots and were only initiated in 2015. It will be interesting to see if different results are obtained after a few more years that the management practices are in place. Okay, now let's get into models. First and foremost, I don't consider myself a modeler, but now I can say I have some experience with models. Models are representations of our understanding of a system to help us better understand real-world problems. In agricultural science, this could represent a myriad of different things, from a whole farm, one barn in a farm, or a single field, depending on what system the model attempts to replicate. If a model accurately represents a system, inferences can be made under variations in management or for predicting the outcome of future scenarios. We know a model is accurate when it has been calibrated and validated for a scenario. Calibration is the process of adjusting model parameters to force model output to agree with real-world data from the process of interest based on goodness of fit indicators. Validation is assessing the performance of the calibrated model on a new scenario using goodness of fit indicators. It's intended to verify that the model is performing as expected. Since models are typically calibrated and validated for a particular use case, they typically come to a local optimization of a particular response or set of responses. Global optimization is distinguished from local optimization by considering all responses. Calibrating models typically finds a local solution for a set of responses rather than the global solution for all model parameters. It has come to our attention that some of these models are sometimes being used in an off-the-shelf capacity without calibration to make inferences about the potential for carbon storage. To that end, the performance of models run in such a capacity is relatively unknown. As we have 3 diverse research sites with excellent soil carbon data, we decided to assess the performance of the commonly utilized denitrification decomposition model in a default condition and after an initial calibration. First, let's look at the DNDC model interface and how data was compared. This is the DNDC model interface. First, climate parameters and weather files are loaded into the model. Then users specify soil parameters like texture and initial soil organic carbon level. Users go on to create a management schedule consisting of crop parameters, planting and harvest dates, fertilizer and manure applications, irrigation schedules, and any grazing or crop cuttings for alfalfa, for example. And once the system is specified, you can run the scenario. The denitrification decomposition model provides some model output on crop performance, carbon, nitrogen, and water balances, as well as estimates of greenhouse gas emissions. However, to assess the performance of the model, we needed to extract the model output and compare it with field observations at the time they took place. For the calibration of the model, we chose to look at biomass carbon, or crop yield, soil organic carbon content, nitrogen mineralization, soil moisture, and estimated crop evapotranspiration. Summary statistics of goodness of fit with field observations were produced. During calibration, model parameters were changed in an effort to optimize the goodness of fit. Although all fit statistics were produced for each treatment in each research site, we simply don't have time to go through them all. Therefore, we will discuss holistic model performance and any issues encountered during modeling. The goodness of fit statistics are shown on the right for overall model performance. The default model condition is shown on the left and the initial calibrated condition shown on the right. For crop biomass, the continuous corn cropping system of the cover crop site was simulated the best, although issues were encountered when trying to calibrate to triticale yield. At GraceNet, the red box in the default condition indicates where 2017 alfalfa growth failed under the default model condition. Initial calibration corrected this shortcoming. However, yield under the first year of alfalfa was overestimated due to problems with how perennial crops are simulated in the model. For all 3 sites, access to soil water had to be improved in order to accurately simulate crop yields. This involved reducing crop water demands and modifying soil field capacity and wilting point beyond their known values to allow more water into the system. This is potentially an indication that water is not simulated correctly by the denitrification decomposition model in our irrigated environment. Cover crop fit with soil organic carbon declined slightly from the default as a result of attempts to better simulate triticale yield. GraceNet fit with soil organic carbon well in the default capacity. However, the calibration efforts improved all fit statistics modestly. The LT manure model in the default capacity underestimated soil organic carbon, probably as the initial estimate of soil organic carbon was low. It was evident at all sites, but especially at the long-term manure site, that the denitrification decomposition model underpredicts nitrogen mineralization where manure is applied at a low rate or where manure is not applied. Okay, that was a lot, so let's pick a few summary points. The soil water module in DNDC did not accurately depict soil water in R3 systems, posing a larger question to similar systems. If soil water is not simulated correctly, there are other effects on decomposition dynamics. Proceed carefully if you intend to use model output such as greenhouse gas values, as they are highly dependent on soil water content. Increasing the nitrogen mineralization by increasing decomposition rates results in decomposition of soil carbon and underprediction of soil carbon levels. Leaving the decomposition rates as default results in crop nitrogen stress, especially when manure is not applied, reducing the accuracy of predicted yields. Soil organic carbon content is hard to model. In the default capacity, the DNDC model's error was over 4 tons of carbon per acre. While calibration can improve the model fit, error was still on the scale of a few tons of carbon per acre. Take care with any absolute values you intend to use regarding soil organic carbon storage. Now, we will conclude the presentation by extending the calibrated models to 2050 and looking at the projected impacts on soil organic carbon. The high and low emission scenarios did not make a difference in the modeling of soil organic carbon levels in any site. At LT manure, there is no projected difference between 8 tons per acre applied annually and 16 tons per acre applied biennially on a long-term basis, supporting our earlier analysis. However, there was no saturation point reached, even under the highest manure application rate. This is concerning for speculations made on potential carbon storage. So the synthetic fertilizer treatment is suggested to slowly mine soil organic carbon in the long term. At GraceNet, fall manure is projected to result in greater soil organic carbon levels after many years. This contrasts my personal opinion, but since the model is based on our observed data, it does make sense given our earlier analysis. Similar to the LT manure site, the synthetic fertilizer treatment appears to slowly mine soil organic carbon in the long term. At cover crop, when manure was applied, no-till did not appear to benefit soil organic carbon accumulation. Within each tillage practice, the treatments with triticale cover accumulated more soil organic carbon when manure was applied. Notably, it appears as though soil organic carbon is projected to decline under only synthetic fertilizer unless no-till is combined with cover crops, seen with a pink line here. No-till is not projected to result in soil organic carbon benefits when manure was not applied. However, keep in mind these are only models constructed with our study's data. Plenty of other research indicates that no-till and winter cover are favorable practices for building soil organic carbon, so don't count them out just yet. That concludes my discussion on soil organic carbon accumulation in our 3 cropping systems, and if there are any questions, I'd love to take them.
All right, great presentation. Does anyone have any questions for Andrew? I guess I have a question. So this was all done on modeling on some research plots. Are there any plans to expand this and look at larger experimental units or ways to validate the model using bigger plots or full fields? You might be muted.
Can you hear me now?
Yes.
Okay. So the denitrification decomposition model can be run in 2 different capacities. It can be run for sites and it can be run for regions. As for now, we haven't discussed running it in the regional simulation, but I suppose that could be one area of interest we could look into.
I was just curious. It does look like there is a question in the chat box. Why was there a lack of soil organic carbon plateau in the modeling? Not part of the model or not long enough time frame or other?
So I can't answer that for sure. I can tell you that when we saw that result, we were curious. So we went ahead and extended out the simulations to 2100. And even by that point in time, plateau levels weren't reached. And it was saying in some of our highest manure application treatments that you could accumulate carbon up into like the 8% range, which we know is just not practical.
Interesting. Yeah, well, thank you for that. If there's any other questions, feel free to put them in the chat box. And would you mind putting, if you're interested, putting your contact information also in there, Andrew? And if anyone has any burning follow-up questions, and I guess we'll queue up this next video, get rolling on the last one for this session. And just as a reminder, this is a little bit of a longer one, taking up two slots, so 40 minutes, but stick around because at the end, we'll have a quick survey and we also have some CEU credits available as well for anyone who needs those credits. Great.
Good afternoon. My name is April Latham. I work with the USDA Agricultural Research Service in Kimberly, Idaho. And today I'm going to be talking about two of our long-term studies that are looking at nutrient cycling related to using manure in both forage systems and commercial cropping systems. So we're going to look at two different long-term studies. The first is what we call the long-term manure study, where we're evaluating manure application rate and timing on nutrient cycling and crop quality. Our second study is what we call the cover crop study, which is where we're evaluating the effects of manure versus fertilizer, different types of tillage and cover crop on nutrient cycling and crop quality. In addition to nutrient management, we also have looked at a wide variety of things on these long-term studies, including soil health, water balances, antibiotic resistance, greenhouse gas emissions, and carbon storage and sequestration. So if you'd like information on any of these other topics, please feel free to contact us and we'll get you the information you're interested in. So the long-term manure study was a study that we started in the fall of 2012, and the official end of the study was this year, right now, 2020. So we have two identical fields, as you can see in the picture on the side here. And the difference between the fields is we staggered the crop rotation. So the north field was wheat, potato, barley, sugar beet, and then in the south field, we started with barley instead. So we have a control where we had no application of manure or fertilizer. We have our fertilizer plots where we applied just commercial fertilizer based on the U of I recommendations for the different crops each year, and 3 rates of manure that we applied either annually or every other year. And this field is located in southern Idaho, so it's a Portneuf silt loam. We have a pretty high pH, close to 8. And everything is fully irrigated because we do not get much precipitation during the growing season. So our treatments, our manure treatments were 18, 36, and 52 wet tons of manure per acre. You can kind of see here a little bit what those treatments look like. Just to give you an idea of what the high manure treatment looks like, this is what it looks like in different situations. People see 52 tons of wet manure, and they think that we're piling the manure on, but actually you can really still see the soil even at that application rate. So these rates were basically picked based on the highest rate being what producers might typically apply in our region, all the way down to a very low rate, which would also require the use of supplemental inorganic fertilizer to meet crop nitrogen and sometimes phosphorus needs. So our goal was to apply the same amount of dry manure each year. It's kind of difficult to balance all your nutrients if we try to do it on an NRP basis. So we decided to do just based on dry weight. And each year we did a pretty good job at the 3 different application rates of balancing for dry weight. But what you'll notice is that due to differences in manure nitrogen and phosphorus content, even though you're applying manure at the same rate every year, you could be applying quite a bit different amounts of nitrogen and phosphorus. And so it's really important when you use manure in a cropping rotation to analyze your manure each year to make sure you know how much nitrogen and phosphorus is being put on. So we looked at first crop yields and quality. So we wanted to know, would crops respond to manure nutrients similarly to what they do when we put on inorganic fertilizer? And were there any negative effects on either crop quality or disease due to using manures? And this is a concern for some of the commodity crops, particularly potato and sugar beet, where you could have soil-borne diseases and pathogens. And then there's also quality concerns related to wheat. So first we'll take a look at barley yields. And just to orient everyone to these sets of slides that you're about to see, what I've done is I've taken and tried to color code them by rate. So the lowest manure rate are in blue, the medium rate is in green, the higher rate is in yellow. And then we move to fertilizer, which is that purple, and the control at the very end. And all the slides will be set up this way, with the annual application being the first bar and the biennial being the second bar within each color. So our barley yield was not affected by manure additions from 2013 to 2017. However, in 2019, the yields at the lowest manure application rate were greater than the yields with a high manure application rate. Although in all of the years, there was no significant difference between whether we used fertilizer or did not use fertilizer. And you can see that at the highest manure application rates, there's a slight depression in yield. And we believe that that's due to lodging, which was a problem in almost every year in the barley at the higher manure application rates. And so we think we're just losing some of that grain in the field during harvest. Of concern related to quality is barley protein. So this is important because as protein is increasing, your malt quality is decreasing. And the barley that we grew was specifically a malting variety. So typically, if you're below 14 or 13.5%, your grain can be taken for malt. However, if your proteins get too high, then your grain will be rejected, usually by the processor, and it'll have to be sold as feed barley, and that's a pretty big loss in dollar value. So we did see that the barley protein content increased with increasing manure application rate in most years. Taking a year off of manure application, so when we applied manure every other year, that did seem to reduce protein content in those treatments. And it was interesting that the barley protein has actually been decreasing every year since 2015. And I don't know if that's just because we're doing a better job managing nitrogen on those sites after we got rid of the background effects, or if that has something to do with climate or some other variable. But I would have initially been concerned about using manure with barley at high manure rates because we were getting to protein levels that would have been unacceptable for malt. However, in the last 2 years, even at our high manure application rates, we were still well below the cutoff threshold for protein. If we look at wheat yields, similar to barley, we did not see a significant effect of manure application rate versus fertilizer. We did see a couple differences just in 2013 between the manure application rates themselves. As with barley, in some years we did have lodging, although in wheat we did not have lodging every year. So in 2019, for example, there was no lodging, and you can see that the yields across the board were very similar, obviously except for the control, which hadn't had fertilizer or any other nutrient source in quite a while. So lodging was our biggest problem with the grain. So you can see on the left, the control always standing up quite well, but our high manure application rates, lots of times the entire plot would just be laid down. And we were actually using a growth regulator. So even when we used the growth regulator, we were not necessarily avoiding this lodging problem. And we believe that's part of why we have the lower yields in the higher manure application rate plots. So one of the things we did notice when using manure is that you tend to have a lot more tillers per plant than when you are just using inorganic fertilizer. And they also tend to have more heads, and we think that the excessive tillering with all those heads on them does create the lodging problem, probably due to a reduction in stem strength. So looking at sugar beet yields, the yields were very similar in every year between fertilizer and the manure application rates. In 2018, the yields for the highest rate of manure were actually greater than for the low manure rates, but they still were not different from the fertilizer. What we did notice, though, is that you definitely have quite a bit more biomass in the tops when we added manure compared to fertilizer. And in lots of cases, the roots were quite a bit bigger in the plots that are receiving manure. And although these differences in yield are not significantly different, numerically they were higher in the manure plots than the fertilizer plots, which will become important in just a minute here. When we looked at the sugar beet sugar percentage, we definitely see a decrease in sugar content with higher manure application rates. And this was significant most of the time. And compared to the fertilizer, you did get depressed sugar content at high manure application rates. However, because you had a slightly higher yield when you had manure application, even though the sugar content was a little bit lower, our estimated recoverable sugar was not different between fertilizer and manure in any year. In some years, there was a slight reduction in the higher manure application rates compared to the lower, but they were never significantly different than the fertilizer. The one other concern is that as you did have higher manure application rates, your brain nitrate in the beets was increasing, and that's a contaminant that has to be removed during the processing of the beets. And therefore, when you price out how much a producer would have gotten per acre based on the recoverable sugar, they could have a slight decrease in price because of an excess of brain nitrate. It all depends on how the sugar companies are doing the calculations in any given year and how big that penalty is. With potatoes, we saw very little effect of manure application on either yield, disease, or quality. The one thing that we did see is that you have, for the higher manure application rates, you did see a little bit lower specific gravity, which could be a concern. But these were not so dramatic that they were a huge concern for us during the project. So looking at what's happening in the soil, because the soil is really going to help moderate and control a lot of the nutrient cycling that we have in the cropping systems, we wanted to take a look and see how soil nutrients were changed by addition of nutrients. And so this is soil sampling that was done this spring, so pre-plant spring sampling in 2020. And so this would have been following 8 manure applications. As you can see, the organic matter increased quite a bit, so our controls were about 1.2%, while our highest manure application rates were at 3%, so more than doubled the amount of carbon in that system. Interesting that the pH actually decreased at high manure application rates, so the manure is kind of acting as an acidifying agent as it's breaking down in the soil. You can see both Olsen phosphorus and potassium increase dramatically with manure applications. So going from 14 parts per million Olsen P fertilizer up to 190 parts per million in the highest manure application rate. And for potassium, going from 150 parts per million in the fertilizer plots to a little over 1,200 at the highest manure application rate. So you're really accumulating quite a bit of nutrients in these soils when you're applying manure. And I did include a few other micronutrients, and as you can see, not only are you getting NP and K when you're applying manure to these plots, but you're also getting a whole bunch of micronutrients in addition to that. One other thing of concern is that the EC and the SAR did increase a little bit, so that's the sodium absorption ratio. And this could be an issue if you have really high manure application rates and you have salt-sensitive crops. So every year we would take these post-harvest cores from each of the plots down to between 4 and 6 feet and look at the nutrients throughout the soil profile. So this is looking at the Olsen P following either 1 manure application on the top or 4 to 7 manure applications on the bottom. And as you can clearly see, continual application of manure at high rates really drove up the soil phosphorus in the first 2 feet. And even though these are really silty soils, high calcareous content, where you wouldn't expect to see a lot of phosphorus movement, even at the high application rates, we're starting to see differences down to 3 feet. So phosphorus is moving through these soil profiles. And that can be a concern around here, as we do have areas where we have pretty shallow soils where you may only have maybe 2 or 3 feet of soil. And if you're continually applying manure at high rates, you could start losing some of the phosphorus to leaching, which could eventually end up in surface waters. But of course, the biggest concern is loss by erosion and surface runoff. And so these high soil test P levels right at the surface could be a big concern for transporting both total P in particulate form as well as soluble P off of these fields. So the agronomic rate is usually around 20 parts per million Olsen, and After our 4 to 7 manure application rates, all of the manure treatments were above what we would consider to be the agronomic threshold. The regulatory threshold has been set at 40 parts per million, and once you get above that, there is some control as to how much manure you are allowed to apply to any given field. And after 4 to 7 manure applications, only one treatment, which was the lowest rate, applied every other year. So only 4 applications. That was the only treatment that was still below the threshold. So it's pretty easy to build up your soil test P, even when you're using what we would consider fairly low rates of application. So it's really important to keep track of how much manure is going on, how much phosphorus is going on, and how much is coming off the field with your crop. The inorganic nitrogen was a little more all over the board, especially in 2013, which is probably due to the residual effect of the past management on these fields. And by 2018, you could see that we did a really good job in the control plots of mining nitrogen out of the soil profile all the way down to 6 feet. All the manure treatments, especially the annual high manure treatments, were quite a bit higher at the topsoil. And even throughout this soil profile, most of the manure treatments were greater than the fertilizer treatments. So we're definitely putting some extra nitrogen into the soil profile, and it is slowly moving down over time. So we wanted to look at how treatments are affecting mineralization throughout the growing season. So we had 2 separate studies, one where we took soils in the beginning of the season and we incubated them in the lab. And then we had a field study where we collected soils from 1 foot and 2 foot depths, and they were put into these buried bags that you can see here in the picture and buried in the field, and then they were pulled out slowly over time. And so that was to be able to mimic the type of temperature conditions that you would have in the field as those bags are impermeable to water. So water is not moving in and out of them, but they are subjected to the same temperature regime that you would have in the field. So if we look at nitrogen mineralization throughout the growing season, you can see that nitrogen mineralization rates are greater for the manure application than the fertilizer, particularly the higher rates of manure application. And that mineralization continues well into the fall. Even when we start to have cooler temperatures, it still looks like it's increasing at the same rate as it was earlier in the season. And this was data from last year, from 2019. And so in 2019, we had grain. And so our grain would have been harvested in August. And so it's important to remember that once you harvest the grain, that even though the crop is not there, you're still having this mineralization happening from the manure in your soil. And so it's pretty important to manage your post-harvest irrigation to make sure that you're not putting on a lot of water after you've harvested to fill up your soil profile for next year, because you could end up moving a lot of your nitrogen out of the profile into groundwater where we don't want it. So this is a look at the net nitrogen mineralization over the growing season. And this would have been the amount of total inorganic nitrogen at the end of the season minus what we started with at the beginning of the season for each treatment. And so you can see here that net mineralization varies quite a bit from year to year. And this is probably driven by climatic variables that would affect microbial activity, as well as it could be related to manure characteristics. Our net mineralization ranged from approximately 25 to 325 pounds per acre per year, with the higher manure application rates typically having greater net N mineralized than your lower application rates, and definitely greater than your fertilizer and control plots. What's really interesting is that our control plots, even though they're not receiving any type of fertilizer at all, they're still producing between 50 and 150 pounds of N per year. So that's quite a bit of nitrogen that's being produced just naturally in our soils. So we compared what we found in the field versus what we would measure in the incubation study, as we're considering the incubation study to be the maximum amount of nitrogen that you would expect to be released. And you can see from year to year the ability of the soil in the field to produce the same amount of nitrogen as that in the incubation studies can vary. In 3 years, we had pretty much the same amount of nitrogen mineralized in the field as we did in the incubation studies, so it pretty much met its maximum potential. But in the 3 other years, it was quite a bit lower. In 2016, it was even as low as 56%, so about half of what we saw in the field. So this gives us a good indication that there's a really big influence on climatic variables in the field that influence nitrogen mineralization. And this also makes it really hard for us to predict in any given year how much nitrogen might be released from these manures. So we looked at the cumulative net nitrogen mineralization over this 2013 to 2019, following 7 years or 4 years of manure application. And we found that the net mineralization ranged from 590 to a little over 1,500 pounds of nitrogen per acre. So that's quite a bit of nitrogen being produced. And our biennial rates ranged between 14 and 40% less nitrogen mineralized than the annual rates. And it was interesting that at the low rates, there was a greater discrepancy between the annual and biennial in terms of how much nitrogen was mineralized over the season. But as you're increasing these manure application rates, the difference dropped quite a bit, down to 14%, which makes me think that we're reaching some sort of maximum mineralization potential at these higher manure application rates. But on average, the annual N mineralized ranged from 84 to 217 pounds of N per acre. And over the same time period, the control plots were releasing on average about 90 pounds of N per acre. So about 30% of the cumulative manure N that was added was mineralized at the highest manure application rates. At the lower manure application rates, it's a little bit closer to 20. But it looks like about 30% is the maximum amount that we're finding over this 7-year time period. So we tried to look at ways to estimate the nitrogen mineralization based on easy to measure soil parameters from samples that were taken prior to planting in the spring. The best relationship looked like it was between the pre-plant total inorganic nitrogen, which is the combination of both ammonium and nitrate, and how much net mineralization there was in the field. And there was about 3.5 times the pre-plant inorganic nitrogen ended up being the amount that was mineralized over the season. Although you can see that this varied from year to year, likely due to climatic events. The other indicator that was somewhat useful seemed to be the pre-plant total nitrogen, and that was not as well correlated as the pre-plant inorganic nitrogen, but was still fairly well correlated. So now we're going to look at the plant nutrient uptake and overall balances in this study. So I'm going to focus on the sugar beet and potato because they were pretty interesting. As we saw earlier, the barley protein increased with manure application rate, and we saw the same trend in the wheat, so there was greater N uptake in both of those crops as well with increasing manure application rate. In the sugar beets, we looked at both the amount of nitrogen accumulated in the tops and in the roots. And what you can see is that the tops accumulate quite a bit more nitrogen than the roots do. And within the sugar beet, there was a significant effect of nitrogen uptake with increasing manure application rate. So when we look at the difference between the nitrogen uptake in the fertilizer versus the highest manure application rate, the highest manure application rate took up 1.3 times the amount of nitrogen than the fertilizer treatment did. And when you look at the amount of nitrogen in the tops, it ranged from about 114 to 266 pounds of nitrogen per acre. And that's important because that residue is going to be chopped off in the field and left and can become a nitrogen source for a crop the following year. The potato vines also accumulated more nitrogen than the tubers did, but they were not as sensitive to manure application rate as the sugar beets were. There looked like there was a slight increase with the highest manure application rate, but not nearly as dramatic as what you see in the sugar beets. And in the vines, again, there was an accumulation of about 137 to 209 pounds of N per acre. And this nitrogen is also returned to the soil at the end of the growing season. So it's really important to account for these additions of nitrogen back into the soil with crop residue that could be used by the plants the following year. If we look at plant uptake over the whole rotation over the 7 years, you can see that the cumulative nitrogen uptake increased with increasing manure application rate. There was a little less accumulated with the biennial versus the annual application rates, but it was only significant at the lowest manure application rate. However, if you look at the difference between N uptake in the fertilizer treatments versus the manure treatments, the fertilizer treatments took up 29% less total nitrogen than the highest manure application rate treatments. So having that manure in the system, especially over longer periods of time, really does amount to a pretty large amount of extra nitrogen that's mined out of the soil versus when you were just using inorganic fertilizer. And so it makes me question, do we need to have different nitrogen removal rates for fields that are receiving manure versus fertilizer? Since this is something that has to be included in our producers' nutrient management plans, maybe we need to account for the difference in fields where you may have manure and have more plant uptake than you would in fields where you just have fertilizer. If we look at the overall nitrogen balance over the 7 years, you can see on the left bar is the nitrogen that was put into the system, and on the right, what's coming out of the system for 3 of the manure application rates, the fertilizer and the control. The nitrogen that's applied with manure is much greater than that removed by the crops. The fertilizer, on the other hand, there was just slightly more nitrogen going in than came out with the harvested crops. However, in the 3 manure application examples we have here, that nitrogen is still sitting in the soil profile if you look at the whole profile down to 6 feet. One of the other losses that you can have in addition to leaching is nitrous oxide losses, and what we've demonstrated on these plots is that we're really only losing about 1% of total nitrogen as N2O to the atmosphere. So it's a pretty small loss to the system. So a lot of this nitrogen is going to remain in the soil. If we take that 30% nitrogen mineralization rate that we saw in the fields, then if you look at what would be the available N versus what was taken out, we were removing about 59 to 112% of that available N that mineralized over that growing period. We'll take a look at phosphorus uptake. Again, this time I'm using sugar beet and potato as an example because they were really interesting to look at from a cropping perspective. In both the sugar beets and potato, the phosphorus uptake was greater in the roots and tubers versus that taken up in the tops. And in both crops this time, there was increasing phosphorus uptake with increasing manure application rate, although this was a little bit greater in the sugar beets than in the potatoes. So phosphorus uptake between the fertilizer treatment and the highest manure treatment in the sugar beet was 71%. And in the potatoes, there was a 40% increase between the fertilizer and the highest manure application treatment. We look at plant P uptake over the 7 years. Similar to nitrogen, the cumulative P uptake increased with increasing manure application rates. However, there was no difference between the annual versus the biennial treatments. And if we look at how much was removed with the fertilizer treatment compared to the highest manure treatment, the fertilizer was 24% less phosphorus removal than the highest manure application treatment. So again, makes me question whether we need to have different P removal rates for fields that are receiving manure versus fertilizer in order to more accurately account for how much phosphorus is being removed. Now we'll look at the overall phosphorus balance over the 7 years, and you can clearly see in the figure that the phosphorus applied with manure was much greater than that removed over the 7-year cropping rotation. The fertilizer P was much more balanced with slightly more in than what was taken out. But even with 7 years of a crop rotation, we've barely removed more phosphorus than what was applied with the first application on those high manure application rates. So that means that this phosphorus is accumulating in the soil. And if you remember back to the soils data, the soil test P has accumulated to approximately 10 times greater than the agronomic rate for these high manure application soils. So P accumulation in these soils becomes a long-term problem, which can have lasting impacts on the environment. It takes a really long time to draw this phosphorus out, and so therefore that phosphorus is available for loss if there's erosion or runoff from these fields. So now we'll switch to the cover crop study. This study is a little bit younger. It just started in the fall of 2015. And this is a continuous corn rotation. And we're looking at 3 different main effects: the effect of having fertilizer versus manure, and the effect of having a cover crop, which in this case is triticale, versus having no cover crop, and looking at a tillage effect of either conventional tillage versus minimum till. And these plots are actually located just to the south of our long-term study plots. So again, they're on a Portneuf silt loam with a pretty high pH, and everything is fully irrigated. And this image was taken from this spring, where you can really see the dramatic effects of the cover crop on the field out there. And those really dark squares are the cover crop where we had manure. The middle dark squares are where we had the cover crop under fertilizer. So again, we tried to match the amount of dry manure that was added. And even when we try and do that, we still end up with very different amounts of nitrogen and phosphorus added. So I will again say this is really why it's important to measure the nutrients in your manure each year, so you actually know how much is going on the field. We look at crop yields. Our main questions were, will the crops respond to manure nutrients similarly to those from fertilizer? And will the inclusion of a cover crop depress the yield of our silage corn? First, we'll take a look at the silage corn yield, which is listed here at a wet weight in tons per acre. And I tried to make these treatments a little bit easier to follow by color coding them. So the first 4 bars are all conventional till, they're in the blue. The second set of bars in the orange colors are the no-till plots. And so we have the first bar in each set being manure with cover crop, the second being manure without cover crop, then no manure with cover crop, and no manure with no cover crop. And the same with the no-till plots. So we can see here there was no effective treatment on silage corn yield in any year, although there was a slight yield depression starting in 2017, which is likely due to the inclusion of a cover crop. And that was because we harvested the triticale in the spring, and the corn gets planted slightly later if you wait for that triticale to mature. And so you lose some of the growing days during the season, which gives you a slight yield depression. The triticale yield, on the other hand, was greatly affected, especially by manure application. So you can see these bars here, the first 2, the bluer ones are conventionally tilled, the green ones are the no-till system, and the first bar in each of those series is where we have manure, and the second bar where we don't have manure. So you can see that the amount of triticale that was produced when you had manure was multiple times that of when we had no manure. There were a couple years, like in 2018, where we couldn't harvest because we got the triticale planted too late the year before, and the yields were too low to harvest. We had the same problem in 2019 with the fertilizer plots. And you can see in the picture below, this is a fertilizer plot next to a manure plot. So the triticale and those fertilizer plots are about up to my knees, but when you go into the manure plots, they're pretty much up to my waist. We look at the total tonnage, though, if we include both the silage and the triticale, there was no effective tillage on these yields, but the fields that had cover crop with manure had a much greater overall yield than treatments without manure and cover crop. And overall, our forage production was increased to about 40 tons per acre when we had the inclusion of the cover crop with manure, though. So even though our silage tonnage was decreased to mid-20s, overall, we still ended up with 40 tons per acre versus the 30 tons that we had gotten before we started implementing a cover crop into the rotation. So even though there can be a yield depression, you can still end up with more forage, especially when you have manure and it's producing that much biomass. We had some issues related to the no-till. We had to switch to minimum tillage, mostly due to compaction, and we were having problems with the planter, so we had to go to strip till. We definitely saw more weeds in the no-till plots, and that accumulation of manure on the surface could be a problem if you have salt issues, which could inhibit emergence, and it could also contribute to greater phosphorus runoff due to surface runoff from the fields, because that manure is just sitting on the soil surface since it's not incorporated. In terms of soil nutrients, we wanted to look and see if those were changing. Our post-harvest inorganic nitrogen, same as with the long-term manure study, we're looking at nutrients post-harvest down to about 4 to 6 feet, depending on the year. So we had increased inorganic nitrogen at the soil surface with manure additions compared to when we did not have manure applied. And we also saw increased inorganic nitrogen with depth for conventionally tilled plots with manure. But we did not see any true clear trends in the effect of cover crop versus no cover crop. Post-harvest Olsen P was a lot more straightforward. You can see after several manure applications that there's a clear increase in Olsen P at the soil surface. And it looks like our no-till plots appear to have a slightly higher P at the surface than our conventionally tilled plots, and that the inclusion of a cover crop actually appeared to decrease Olsen P at the surface compared to not having a cover crop. So we'll look at the plant nutrient uptake imbalances in this study. Plant nitrogen uptake over the 3 years, the cumulative uptake was greater when there was manure and cover crop, of course, because the cover crop pulls out an additional amount of nutrients. However, there was less of an effect on the cover crop when you just had fertilizer. It was that huge boost in biomass that we got with the addition of manure that was generating these larger uptakes of nutrients with the triticale. There was more nitrogen uptake with the manure fertilizer, with manure versus fertilizer, even when we didn't have a cover crop. So the effect of manure just on the corn was apparent like it was in the other long-term manure study. And tillage had no effect on nitrogen uptake. This reinforces the need to have different removal rates for fields that are receiving manure versus just fertilizer. If we look at the overall nitrogen balance over the 3 years, you can see again nitrogen in on the left, nitrogen out on the right for each of the treatments. So we have conventional tilled, which is the C, N is the no-till, M for manure, NM for no manure, and T for triticale, and F for fallow. So obviously, again, like we did with the long-term manure study, you can see that the amount of total nitrogen that's going onto these plots when we add manure is pretty significant and quite a bit greater than the nitrogen that's coming off. And in this case, even after the 3 rotations, the amount of total nitrogen that we put on with one application was pretty much greater in almost all cases than the amount that was pulled out over those 3 years. In the non-manure treatments, there was slightly more nitrogen out than in each year, but that does not account for natural nitrogen mineralization, so we were never deprived of nitrogen in this study. If we assume again a 30% nitrogen mineralization rate across years, then the nitrogen removed was about 93 to 130% of the mineralized N. So obviously we're picking up some of the control natural mineralization in some of these studies to help meet the crop needs. Plant P uptake was similar to what we saw with the long-term manure study. So there was greater uptake with manure addition and cover crop. But even when we did not have cover crop, there was still greater phosphorus uptake over the rotations when we had manure versus no manure. And again, tillage did not have any effect on uptake. So again, this reinforces the need to have different P removal rates for fields receiving manure versus the overall P balance on these sites after the 3-year cropping rotation can be seen here. And you can see that the phosphorus applied with manure was much greater than that removed in those 3 years, whereas the fertilizer was pretty similar between what was going in and what was coming out. So this phosphorus has accumulated in the soil surface again, and the soil test P now is approximately 10 times greater than the agronomic rate when we had these manure applications. So even with one manure application, we added more phosphorus than what was removed with this 3-year cropping rotation. So you can see, again, it's really easy to build phosphorus up in these soils, and it's pretty hard to draw them back down once you've gotten to really high levels. And that's all we have time for today. Thank you so much for your attention, and I'm here to answer any questions. And if you're listening to this at a later date, please feel free to email me at my email address here on the screen.
Great presentation. If anyone has any questions, feel free to put those in the chat box.
They all went to lunch, Linda.
Yeah. Probably not. I don't know. I mean, yeah, this difference in time zones is, I think, I wouldn't say rough, but especially those that are not necessarily in Pacific time.
April, thanks for providing all of that information. It was really, really nice to see. Your site was one of the ones that we sampled in our project, and you have a lot of data that is supplementary. So I would love to, maybe this is something to chat about later, but I would love to talk to you about how we might be able to work with the supplementary data that you have to help with where we're trying to go with our analysis.
Absolutely. You can have whatever data works for you.
Okay, well, I've got a list of things that I have been wishing we measured. We measured a lot of things, but you can only do so much when you've got 2,000 samples. So I'll get in touch with you soon.
Okay, great.
While everyone's thinking about questions, I think I'm going to launch this poll that you all can fill out. And then after the poll is done, then I will put up the sign-in sheet for the CEU credits. So I thought your presentation was really interesting. I guess what's next? You said it was for LT, it's an 8-year study. Do you know what the next years are going to look like?
Yeah, so in that long-term study, since we've built the soil nutrients up so high, our next goal is to do kind of a nutrient drawdown study and look at 2 different cropping rotations. So we're going to look at a corn triticale rotation, which is pretty typical for our dairy producers. And then I'm going to do an alternative forage rotation that'll be corn, followed by a forage barley canola mix, followed by sorghum Sudan grass. And we're going to try and see how long it's going to take to draw phosphorus back down out of those soils, because that's one question that our dairy producers have. And also look and see what the residual effects of nitrogen is in that system and how much nitrogen mineralization we're going to be getting even after we stop applying manure. So that's going to probably run for the next 7 or 8 years at least.
That's fantastic. While we continue to let this poll run, I do have another question if no one else does. If you could go back and kind of restart this study all over again, is there anything you'd do differently? And you don't have to answer either.
Yeah, think about that. It was a lot of treatments. I don't know if there's anything I would do differently except maybe not plant potatoes. The potatoes were hard and they weren't as responsive to the treatments as some of the other crops, although I think the data that we got is really useful for producers who would be interested in planting potatoes on fields that had a history of manure application. Because that's one thing that, you know, in our region in the past, most people haven't had to deal with that. And so I think that some producers were a little bit worried about what negative impacts having that manure could be on potato production. And we didn't really find much of anything, which is great from a production standpoint. So producers shouldn't be worried about having potatoes in a field that's received manure. But from a scientific standpoint, I guess it's not quite as exciting when you don't have much of anything to say. The potatoes love the manure, everything was great. We didn't have disease problems or pathogen problems on the potatoes. So it ended up being a really great source of nutrients for potatoes in our region.
Great. Does anyone have any other questions? I feel like I'm always the one who's asking them all.
That's your job as moderator.
Everything's just so interesting. I'm going to share the results of this poll in case anyone is interested in looking at that. It seems like it was valuable and most people would recommend the event and would like to attend. Yeah, I think that's all we've got for this session. I will share the CEU credit sign-in, which you can just use the QR code to sign up for those, and watch the website because we'll have plenty of, all these videos will be uploaded eventually to the site, and there's still a session going on for the next hour, I believe, on technologies related to manure and byproduct management. And tomorrow we have another full morning and then also some case studies in the afternoon. So I appreciate you all coming to attend this, and let me know if you have any questions. I'll throw my email address into the chat box if anyone wants to follow up with anything. Yeah, thank you all for attending.
Linda, how long will the presentations be available to people who are registered to the meeting?
I think that the presentations are going to be available long-term. I don't think that they have any intention to pulling it down. And since it was a free conference, they should be available for anyone who wants to look at them. I'm not sure what the timeline is to get them all uploaded back to the website. That would be a good question for Joe Harrison with Washington State, who I'm sure you've all received emails from.
And he would be the one to tell us what the website is, like where those will be hosted.
I think it'll be, he will follow up, but I think it will be hosted on the main conference website.
Okay.
Where you can find the agenda and where you registered and things like that.
Okay, thank you.
Yeah. All right, well, I will leave this CEU up for a little bit just to make sure that everyone can get it, but I will close out this session, and thank you to all the presenters who talked about their really interesting studies today. Have a great rest of your day.